Hi everyone, today I'm going to explain to you as fast as possible the cyclic AMP pathway. Cyclic AMP or cAMP is a secondary messenger which is being made in order to transduce signals from the primary messenger, which can't enter the cell due to their chemical structure. Such primary messengers are hormones like adrenaline, glucagon, vasopressin, follicle-stimulating hormone, also neurotransmitters and other type of molecules. Let's say that you haven't eaten in a while and the body didn't get nutrients and the glucose level is low. In this case, glucagon will be secreted by alpha pancreatic cells in order to raise the glucose level in the blood. So we have a nice little glucagon molecule here, which is a ligand because it binds to a receptor. So first, it will bind to its specific receptor, which is a G protein coupled receptor, abbreviated GPCR, and colored in red in this diagram. Why is it named so? Because this receptor is bound to a G protein. And this G protein is bound with a guanine nucleotide, and therefore the name G protein. This protein is made of three subunits, alpha, beta, and gamma. And in its inactive form is bound with a GTP molecule, and in its active form with a GTP. So whenever the ligand binds to its receptor, conformational changes occur within the receptor, which lead to conformational changes in the G protein and the exchange of GDP to GTP. Once the GTP is coupled with a G protein, it is activated and step 3 occurs. The alpha subunit is now freed. When it's free, it will activate this green membrane-anchored enzyme called adenylate cyclase. When it's activated, it will start converting ATPs into cyclic AMPs by removing two phosphate groups and connecting the remaining phosphate groups to a 3' carbon-bound oxygen and giving it structural cyclic appearance. More and more cyclic AMP is produced, and once it reaches high enough intracellular level, it activates the next very important enzyme called protein kinase A. This enzyme is a kinase, so what does it do? Right, it phosphorylates other proteins. By phosphorylating, it makes them more active or more passive, which means it can both activate and inhibit them, depending on the specific protein. Some proteins may be more active in their phosphorylated form, some might be inhibited. It all depends on their unique structure. For instance, continuing our glucagon activation of cyclic AMP pathway, the protein kinase A will phosphorylate specific proteins involved in glucogenolysis and glycolysis. Glycogen phosphorylase will become active and glycogen will start being cut into glucose 1-phosphates, so we can raise the concentration of glucose. But on the other hand, pyruvate kinase in its phosphorylated form is inhibited, which will lead to the decrease of slices of glucose, so more glucose gets preserved and therefore achieve the needed result of raising glucose blood level. All of these phosphorylations lead to metabolic changes within the cell and organism. Remember that not only glucagon activates the cyclic AMP pathway, but many other hormones do. This diagram holds true for its every aspect, only the ligands and the phosphorylated proteins by protein kinase A are variable. The rest stays the same. For today, I was your host Bulgarodan. Stay healthy and leave recommendations for the next video topic in the comments below.